As we wrap up 2021, the Governor General of Canada awarded 3 British Columbians with the Order of Canada for their work in biomedical research. A remarkable acknowledgement for Peter Cullis, Connie Eaves, and Peter Zandstra, all of whom are playing an increasingly important role in establishing Vancouver as a hub of exceptional research in biomedical engineering and life sciences. Zandstra, Eaves, and Cullis are world-renowned scientists pushing the boundaries of research in drug development and stem cell technology. Dr. Zandstra says, "I'm excited to see biomedical engineering being recognized for its current and potential impact in Canada and abroad." Dr. Zandstra goes on to say the appointments of Dr. Connie Eaves, a giant in stem cell and cancer therapy, as well as Dr. Peter Cullis, one of the key scientists behind our ability to use mRNA technology in vaccine development, shows the impact of interdisciplinary research and education and projects to the leadership that UBC's School of Biomedical Engineering is building at the interface of engineering and medicine. Dr. Zandstra is recognized for more than 20 years of groundbreaking work at the intersection of biology and engineering, discoveries that have facilitated the development of technologies for stem cell-based therapies that are used for regenerative medicine and furthering the understanding of stem cell biology, immunology, and tissue regeneration. I invited Dr. Zandstra to join me for a conversation that matters about the role his research has played in finding cures for debilitating chronic diseases and the greater biomedical engineering and life sciences community in Vancouver and British Columbia. Dr. Zandstra, welcome.
Thank you very much. Pleasure to be here.
You know, for a lot of people, they go, biomedical engineering, I don't get it. Like, what exactly is that, and what role is it going to play as we move towards much more precise and, well, designer medicine, the kind of work that is going to be tailored to you, the patient?
Yeah, biomedical engineering is actually a really broad field that has a long history. And I think what's very exciting is that history now is impinging with our increasing understanding of how cells, molecules, and tissues function and come together. And what we're doing is we're seeing for the first time the ability to take engineering sciences that traditionally have had a huge impact in things like pacemakers or hearing aids or vascular stents, and start to have an increasingly large impact in molecular medicines and cellular medicines, because we can take the engineering principles of how systems and signals work and apply them to living systems. And this is really what we're trying to do at UBC in the School of Biomedical Engineering, is bring this engineering design philosophy together with fundamental biology to really important biological problems.
I have 1,000 questions about this because I start to think of the myriad complications that go into saying, well, how am I going to direct that cell to be able to do this? How do you go about being able to do that in a way that is also going to be responsive to the individual patient?
Yeah, I mean, it's a great question. We're very much still at the starting point for that, but we can start, I would start, probably the best way to think about it is you start with a molecule or a protein. And that we can start to design in a computer in silico about how it will come together, what its binding properties are. And of course, that molecule will then, it becomes part of a larger system, a system of molecules and eventually cells that can start to guide the signals that happen biologically towards certain outcomes. One example that I'll give is that is being developed here in Vancouver is when we put specific proteins on the surfaces of T cells, which are cells that can attack and kill cancer cells in a targeted way. Those so-called engineered chimeric antigen receptor engineered T cells are examples of the types of signals which we can put on cells and start to get them to target certain types of diseases in a very personalized manner.
Okay, how did we get to this point that we're able to now model that in computers? Because until there had been recent advances in computing technology and engineering, we weren't able to do that. We literally had to go into the lab and go through a trial and error process. What happened in computing technology that allows us to do that? And I'm going to sort of double barrel this question. And what happened in Vancouver that we seem to be leading the charge in this area of research and development?
Well, it really is a foundation that is based on the genomic revolution that happened in the 2000s. So over time, we've learned about what the parts of cells are. The Human Genome Project identified many of the genes that are there, and that we understand how those genetic components start to be expressed and related as cells change their identity. And that starting point gives us a way to identify the parts list, or what are the parts of a biological system that we can start to engineer. That really represents a starting point for how then higher order structures might emerge out of these biological systems as genes make proteins and other types of molecules out of those systems. So, although we're very much at the starting point of what we can do in terms of engineering design, we're quite far ahead of where we were even 20 years ago. In terms of the other side of your question, what makes Vancouver so special? Vancouver has a strong history of developing a number of different types of technologies. In the 1980s, there were companies started by people like Julia Levy and others, which created an innovative environment that helped create a knowledge base, which then went out and propagated into other companies, which then emerged over the last little while. And that ecosystem that we're creating between the biotech companies in the Vancouver ecosystem and the universities that we have here is starting to create a critical mass that I think is very unique in Canada, and already emerging to be amongst the most exciting ecosystems in North America.
So you are, of course, connected to the Michael Smith project research and so on. How important was his role in helping to establish the ecosystem that allows for Vancouver and British Columbia to start to become this center of excellence? And not just in British Columbia, but I think globally we are establishing a reputation here on the west coast of Canada.
Yeah, I mean, Michael Smith was really a giant in Canadian science and international science. Of course, he won the Nobel Prize for site-directed mutagenesis, which is the ability to change specific sequences in protein coding sequences and change the engineering of those proteins. And that's a very good example of the starting point of that type of engineering design. That type of scientific leadership, and you see that as well in people like Peter Cullis and Connie Eaves, really both attract good scientists and their students to Vancouver, but also attract investment, which is so important for generating continual growth in the ecosystem and the types of companies which are formed around us.
So coming out of that, of course, that work done by Michael Smith, we saw the development of Genome BC, and we know that Dr. Ricks played an important role in supporting that. But at the same time, I can't help but think that the influences of Terry Fox and what was happening through cancer research kind of merged perfectly to help to accelerate the growth of this great network that is coming together here.
Yeah, I mean, there's no question that the Genome Sciences Center, which emerged, the Terry Fox Labs, the Michael Smith Laboratories, all and indeed, research at UBC and in other universities throughout British Columbia, created this kind of molecular foundation that allows us to really be leaders. I think what we're trying to do now at UBC with the School of Biomedical Engineering is build on top of this foundational information and start to think, how can we use these tools and these technologies to start to design either new solutions to disease, or to uncover fundamental mechanisms about how cells make decisions. Some of the faculty that we're hiring within the school now are developing new ways of editing sequences inside cells. Others are developing new ways of delivering and engineering lipid nanoparticles so they go to specific places in the body. So there's really a lot of exciting stuff that's being put forward from this foundation that was created through these many years of research in British Columbia.
Because I'm wanting to sort of give people who are still kind of new to the topic a little bit more of the foundations or contributors that have led to this great environment that we have. We cannot overlook the contributions of Dr. Connie Eaves and Alan Eaves and stem cell technologies. How important is that? Because when I take a look at your website, you talk about that you're working with somatic and pluripotent stem cells to be able to do the work that you're doing. How much does it add to having such great resources in both Dr. Alan and Dr. Connie Eaves here in Vancouver as well?
It's absolutely fantastic. I did my PhD with Connie Eaves, so I learned from her and Jamie Perret about the fundamentals of blood stem cells or hematopoietic stem cells. Connie has really been a leader in the field in defining properties of these cells and helping to quantify how we measure those properties. With Alan, they have put Vancouver on the map with respect to stem cell technologies, really through both the companies they've created and the work that they've done. So it's we're really lucky to be able to work with and build on the science which they've created in the city.
And we have now created a community. When I did an interview with Dr. Mark O'Mara, he said, this is why he came back to Vancouver. Even though he was educated here, he had gone away because he felt that the really important work was being done elsewhere. But now he's come back here because he reaffirms your position that there are remarkable advances that are taking place here. And we've now created a community where we're drawing more and more talent, such as yourself, into Vancouver to push forward. So now let's talk a little bit more about what your work is. I was watching an interview with you and you were talking about the recapitulation of blood development, and I can't help but say, okay, why is that important? How do we do it? And where do we go to keep building upon that work?
So let me tell you a little bit of what my lab does. I guess, first of all, in terms of background, I was attracted back to UBC about 5 years ago through this opportunity of starting the School of Biomedical Engineering, and this foundation of starting to think about molecular and cellular systems from an engineering perspective is really an opportunity that we're building on. So my lab is interested in how cells make decisions, and specifically how we can take blood stem cells either derived from patients or blood progenitor cells, which are generated from pluripotent stem cells, which are a type of stem cell which can turn into any cell type in the body, and generate functional blood cells from them. This is done by mimicking the environment around those cells to try to recapitulate the molecular and cellular signals which are important for the development of those systems. And we've applied them to a number of different applications. Growing blood stem cells has been useful to create new therapies that are currently under clinical investigation for diseases such as leukemia. And in the pluripotent cell space, we're generating immune cells that we hope one day will be useful for targeting specific cancers. So as bioengineers, what we do is we try to design bioreactors, in fact, which are environments where we carefully control and recapitulate this environment to try to get convince the cells that the signals they're getting are very similar to the signals they would get inside the body, and thus undertake these normal developmental steps.
So with these pluripotent stem cells that then become whatever we need them to be, will they self-replicate? Or is this going to be, are we still at that stage where we're looking at ongoing treatments or, application to the patient?
Yeah, so pluripotent stem cells are unique because we can grow them in unlimited numbers. However, as the cells themselves, they're not useful clinically. We have to what's called differentiate them or turn them into different cell types. And there's companies across the world that are working with pluripotent stem cells to generate different cell types, cardiac cells, cells of the blood, of the brain, the neural cells, or cells of the blood forming system. What we're interested in doing is understanding how to connect the pluripotent stem cell to the blood forming cell through a series of steps called differentiation steps. And once we can do that, those cells themselves form the therapy in this particular case. And that would be a therapy that could be used once, and then that treatment would either allow those cells to remove the disease, or if it was a somatic cell or a non-pluripotent stem cell that was used, that therapy could be transplanted and the progeny for that cell would be there in the patient for a number of years.
Okay, explain to me what the fourth pillar is and why it's so important, and what position we're in right now with regards to this.
If you think about the different types of therapies or drugs that we can use, there are chemicals. So chemicals, aspirin or other chemicals that we can design is one example of that. There are proteins is another one, is another type of therapy. So erythropoietin is a good example. There's other antibodies that are used, and then there's more complex combinations of proteins and delivery systems such as lipid nanoparticles, and really living cell therapies are the fourth pillar of medicine. And this is an example where we start to use these cells as tools to treat disease or to cure disease based on replacing certain functions which are currently not available anymore in the patient. This could be using cardiac cells to treat cardiac disease. There's a clinical trial ongoing in Toronto from a company called Blue Rock Therapeutics, where they're using dopamine neurons to treat Parkinson's disease. So these are examples where we're at the very beginning of using living cells as therapeutics to treat degenerative diseases, and it's a very exciting and growing area.
Okay, how long before this moves beyond the research and lab stage to actually being available to patients? We've looked at the promise of having mapped the human genome and been expecting and hoping for bespoke medicine to the individual patient, and still there's so many elements that have to fall into place. Where are we at in being able to take this fourth pillar, as you say, these living cells, and be able to deliver them to the patient, to the time when they can actually go into the pharmacy and say, that's what I need?
Yeah, it's a great question. Well, what's exciting is that there are commercially approved products that are based on living cell therapies available right now, both in Canada and across the world. An example of this are immunotherapies or T-cell therapies, but there's really big problems with these right now. One of the problems is that they're incredibly expensive. So one of the things that we're trying to do as engineers is to think about better ways of manufacturing these technologies to reduce costs, to increase efficacy, or even to find alternate solutions to activate these cells where the amount of manipulation that's needed outside the body is much lower, and therefore the cost and ability to deliver it to many people and make it accessible to the people that need it has a greater probability. So to answer your question really clearly, these therapies are available. They're being used across the world for a number of different diseases. Many, many more are in clinical trials. It's estimated there are many thousands of clinical trials underway in Canada, the US, and Europe using living cell therapies. But we're not at the stage where we figured out how to make this a truly accessible and off-the-shelf technology. And that's really something we're working on very hard right now.
So then, sort of coming full circle in our conversation here, what do we need to do next to be able to bring those next advances into this discipline, biomedical engineering, and then also to be able to do it here in British Columbia?
Absolutely. I think many of the pieces are starting to be put into place for this, but we need to continue to grow. This is not an area that the world is sitting back and just watching happen. There's high competition for talent. So the types of things that we're trying to do at the School of Biomedical Engineering with training the next generation of biomedical engineers with knowledge base in genomics, cellular and molecular engineering, and other aspects of biomedical engineering is really important. I think the other thing we need to do is start to bring, and you've heard about this during the pandemic as well, the ability to manufacture and test at research scale in clinical trials these types of technologies as they emerge out of our universities is also really important, because those create sticky technologies that stay in the ecosystem, that grow through early companies and then eventually scale. So the ability to have facilities and infrastructure to test those technologies is also something that's very important. And finally, we need people. We need to continue. And one of the things we're trying to do at the School of Biomedical Engineering is hire faculty and recruit students that can really participate in the many opportunities that are now in the Vancouver biotech industry.
It still comes down to people. It comes down to brain power, which of course you, Dr. Cullis, Dr. Eaves, and so many others bring to this community. Let me congratulate you on your induction into the Order of Canada, and thank you for the tremendous work you're doing here in Vancouver, out at UBC.